My name is Sabdat Ekama and I'll be presenting this video abstract on behalf of the co-authors on the article titled Mucoadhesive Microspheres of Maraviroc and Tenofovir designed for pre-exposure prophylaxis of HIV-1 and in-vitro assessment of the effect on vaginal lactic acid bacteria microflora. Heterosexual intercourse is a major mode of male-to-female transmission of HIV-1 infection, and women account for a greater percentage of HIV prevalence globally. This has necessitated the development of pre-exposure prophylaxis measures such as microbicides as prevention strategies. Previous microbicide formulations had challenges to its use such as short residence time in the vagina, leakage, and coital-dependent use. This has led researchers to the development of targeted drug delivery systems such as microspheres that can target the drug to the site of action and improve vaginal residence time. This study aims to formulate and evaluate microspheres of the antiretroviral drugs Maraviroc and Tenofovir intended for a candidate vaginal microbicide and assess its effect on the vaginal lactic acid bacteria microflora. A full factorial experimental design at 2 levels of concentration requiring 2 to the power k runs was used to determine the varying effect of independent factors such as concentration of the polymer, the crosslinking agent, and curing time on dependent variables such as particle size, mucoadhesion, and encapsulation efficiency. Maraviroc and Tenofovir microspheres were prepared using Ionic gelation technique, in which the polymer containing the antiretroviral drugs were cross-linked with sodium tripolyphosphate to form microparticle carriers for the drugs. Varying weights of chitosan were dissolved in 10 ml of 4% acetic acid and stirred using a magnetic stirrer for 30 minutes with subsequent addition of 10 mg of the individual antiretroviral drugs. The resultant dispersion was collected in a syringe and added dropwise into a solution of sodium tripolyphosphate to achieve cross-linking. The gel-like bits formed were allowed to stand for a curing time of 15 to 30 minutes, which is the wet microspheres that we can see, and subsequently it was decanted, washed with milky water, and dried at 45 degrees for 12 hours, which is the dried microspheres that we can see. And the microspheres were viewed from the microscope as seen in plate 1b. Mucoadhesion test using sheep vaginal tissue was used to evaluate the mucoadhesive ability of the formulated microspheres. Sheep vaginal tissue was obtained from a freshly slaughtered sheep in an abattoir. The vaginal tissue was cut into the size of a glass slide and rinsed with prepared simulated vaginal fluid and attached to a glass slide. The microspheres were counted and 30 pieces were attached to the vaginal tissue, which was subsequently immersed into a beaker containing 900 ml of the simulated vaginal fluid. The slide was attached to the arm of a disintegration apparatus and given a vertical reciprocating movement for approximately 30 cycles per minute. At the end of 1 hour and at the end of 12 hours, the number of microspheres that were still attached to the tissue were counted, and the percentage mucoadhesion was calculated by expressing the value as a percentage of the originally attached number of microspheres. Drug release studies was carried out according to the method of Agroholi et al., in which the formulated microspheres were each resuspended in 3 ml of a mixture of seminal fluid simulant and vaginal fluid simulant, and the dispersion was transferred into a 10 ml capacity dialysis membrane. The membrane was maintained in a 20 ml release medium contained in a dialysis tube, and the whole system was placed in a conical flask, then placed in a shaking water bath, and the temperature was maintained at 37 degrees. At periodic time intervals of 0 hour, 1 hour, 3 hours, all through to 72 hours, 1 ml sample was withdrawn from the release medium in the dialysis tube for HPLC analysis. And at each point of withdrawal, an equivalent volume of the release medium was used to replace the one that was withdrawn to maintain sink condition. The amount of Maraviroc and Tenofovir released was determined using HPLC analysis. An ideal microbicide should not disrupt the activities of vaginal lactic acid bacteria, which helps to maintain the protective acidic pH of the vaginal environment. Lactobacillus assay was therefore carried out to evaluate the effect of the microspheres on isolated vaginal lactic acid bacteria. Ethical approval was obtained to conduct this study, and it was carried out according to the Declaration of Helsinki. The women gave consent to participate in the study, and vaginal samples were collected using sterile vaginal swab sticks, and the lab organisms were isolated on MRS agar. DNA extraction was carried out using the Gina Bioscience DNA extraction kit, and polymerase chain reaction was carried out using the lab-specific primers. These primers produced an application size of 526 base pair by all lactic acid bacteria. The amplified PCR products were purified using the Gina Bioscience purification kit, and it was sequenced for the 16S RNA gene, and the corresponding sequences were identified using the online blast search and submitted to the GenBank for gene accession number assignment. The identified organisms were Lactobacillus fermentum and Enterococcus faecalis. An MTS assay was carried out in which the organisms were exposed to the microsphere suspension for 48 hours, after which a microplate reader was used to determine the absorbance, and the percentage viability, which is a measure of the number of live cells, was determined. HIV efficacy testing was evaluated against the HIV-1 BAL viral strain. TZM-bl cells were plated into 96 well plates, and 100 microliter of serial dilutions of Maraviroc and Tenofovir microspheres were added to the wells. Nonoxynol 9 gel was used as the negative control, and the plates were incubated for 48 hours, after which the medium was removed and replaced with 100 microliter of bright glow, and the luminescence was measured, after which a dose response curve was used to describe HIV infectivity rate. The results are as follows. Table 1 shows the predicted and experimental values of the optimal conditions of microspheres obtained by numerical optimization. Numerical optimization was used to identify the conditions for the optimized formulation and a prediction of the outcome variables. The two sets of values shows adequate proximity between the experimental and the predicted values. The optimal formulation had moderate particle sizes, high percentage mucoadhesion, and good encapsulation efficiency. An analysis of the in vitro drug release for Maraviroc and Tenofovir from the microspheres was carried out by fitting the release data into the various kinetic models to determine the coefficient of correlation. The model with the highest coefficient of correlation best explains a drug kinetic release pattern. Maraviroc release followed a zero-order kinetic model and a super case 2 drug transport mechanism, while Tenofovir release followed a Higuchi model release kinetics and a non-fictional drug transport mechanism. Results from the Lactobacillus viability assay shows that Lactobacillus fermentum, after a 48-hour exposure to the microsphere suspension, had a percentage viability of 93.9%, while Enterococcus faecalis had a percentage viability of 89.7%. There was no statistically significant difference between the absorbance of L fermentum and the negative control, and the absorbance of E. faecalis and the negative control. But there was a statistically significant difference between the absorbance of the organisms and the positive control. And this shows that the organisms were able to thrive in the presence of the microspheres. The pre-exposure prophylactic ability of the combined optimal Maraviroc and Tenofovir microspheres were evaluated via an in vitro concentration response procedure using nonoxynol 9 as control. The HIV infectivity of the combined microspheres compared with nonoxynol 9 showed a marked difference in HIV infectivity rate against the HIV-1 BAL virus strain. The observed continuous decline in HIV infectivity of the microspheres indicates adequate release of the drugs from the polymer and anti-HIV activity. In conclusion, the antiretroviral drugs loaded in the microspheres had good mucoadhesion, which is a potential for prolonged residence time in the vagina. The antiretroviral drugs were adequately released from the microspheres and showed efficacy against the HIV-1 BAL virus strain. There was no significant disruption in the growth of lactic acid bacteria, which constitute the valuable bacterial microflora of the vagina. Thank you all for your attention. This project is supported by the Fogarty International Center of the National Institute of Health under award number D43TW010934. The content is solely the responsibility of the authors and does not necessarily represent the official views of the National Institute of Health. Thank you.